Hello again, everybody. It is time for Q&A with A&V. I'm Vincent for Racaniello, and right next to me, I guess it would be on this side, is Dr. Amy Rosenfeld, and she is in her lab at Columbia University. Hello, Amy.
Hi, Vincent. How are you today?
I'm well. How are you doing?
Good.
Assays going well?
Well, they hit a little snag when the lady contaminated all the bottles. But so I want to shout out to the people who I encountered walking down the street when we were walking Gorda with my mom and Wendy on Saturday in New Jersey. So there are like 10 listeners all in Maplewood. So that's kind of cool. And also, I keep forgetting to say thank you to the woman who sent me the beautiful 3D postcard from France.
Nice.
That's so nice.
Amy, did you notice I got a haircut?
I see you got a haircut. You got more than one cut. Yes. Very good.
Haircut. I went to this person I've been going to for many years, and she was very excited. She got her first Pfizer shot today.
Excellent. I got Pfizer too.
So listen to this. Her parents live outside of Shanghai in China, and she said their town has had 0 cases the whole time.
Well, how many people are in her town? 2?
No, not 2. But, you know, not every place in China got hit. They did a good job at restricting the spread. So,
well, they made it so you can travel within the country. Yeah, I think it's great. And when are her parents going to get vaccinated?
She said they don't want to be vaccinated.
Well, that was the wrong decision. So we can re-ask them, and maybe they can learn to make the right decision.
Yeah, do you know why they said that? Because there are no cases. Although last night I learned on China TV that there are 6 new cases in the past week, and there hadn't been some for a while. But her parents don't want to be vaccinated because there's no infection here. Why should we be vaccinated? So that's not a good view.
Wait a minute.
And I told her you need to talk to them.
Yeah, would that be my line about why the boosters were like a month apart versus like a scientific decision? That's the argument I've always made, right? Why is there a little echo now? I don't like it.
Where's the echo?
I don't know.
Sometimes
now I hear email binging, or maybe that was my email binging.
On the other side of the room?
Yeah, maybe.
Yeah, it's
maybe
not
it was my computer.
So let's take some questions here.
Okay.
First of all, mother received the second, started feeling crummy, took 2 Tylenol, took 2 Advil. Any concern from dampening from the pills? I think
no.
No. Last week, Amy said no worries, and so did Daniel Griffin.
Yeah, Daniel says not a problem.
All right, now Pfizer says the vaccine is good in 12 to 15-year-olds. So here's what Amy sent me today.
From the Wall Street Journal.
Wall Street Journal. So we don't have the data, but let's see.
Sure
what did we do here?
So I heard that they are submitting it as soon as possible. They're writing it up.
2,200 children, no safety concerns, 100% effective in preventing symptomatic disease. 100%. Now, people are saying, why was it so fast? Because a lot of cases still, and this is a small trial.
Yep.
And so that's really good. Now, I'd like to see the data, wouldn't you, Amy?
Yeah, but I'm giving them time to write it up. You know, it takes a little time to write up the paper. It takes us a while to write up the paper. What was back to back was the safety for pregnant women and the transmission of antibodies to newborns.
Mm-hmm. Where was that?
It was also in the journal, but it's also in some article.
See, I don't subscribe to the journal, but Amy does, so I depend on her to feed me Wall Street Journal stories. Although, Amy, did you know I was interviewed the other day by a Wall Street Journal writer?
I did, Amy Marcus, right?
Yeah.
You talked about spillover events or something, right?
And today I was interviewed by a writer from The Street, And he was impressive. He had read my blog. He'd listened to podcasts. He listened to lectures and was quoting from them.
That's great. And
Oh my
we
gosh.
did concur. What did he concur?
I don't know what he concurred, but we talked for 45 minutes, and he said, I understand that Tommy Thompson calling anthrax a virus is what you got started in communication. And I was blown away by that. All right, Alan wants to know, why did the children's trial need placebo when safety rather than efficacy is the thing, placing the placebo wing at risks? Well, it's an efficacy trial. You need to know if the vaccine works, and you also collect safety data at the same time. Right, Amy?
Right. They're not doing phase 1, phase 2. They're just doing phase 3s to expand the usage of the vaccine.
Hello from Gaza. Is there any advantage that AstraZeneca chose not to use prefusion stabilized? I can't think of any because it's not difficult to add 2 prolines, right? I just think they didn't think of it.
It's hard for me to believe that they didn't think of it. It was out there for a long period of time. They just chose to be different for whatever reason. I'm not going to guess.
All right. Why do I feel like gain of function research is a good thing?
It is a good thing.
To get infected by mistake, has this happened?
So this is a problem that people seem to think that lab accidents happen all the time. Have you ever heard of a lab accident of gain of function? I'm not aware of one.
No, someone being affected? No, never.
Right. And
very careful. I look at BSL, as you know, for being trained in a BSL-3, you have to keep records of every accident, right?
Yes. But the other thing is I just look at our BSL-2 and I have young people and inexperienced people come in and out all the time. And we've never had an accident. And I don't think that the lab that you got your PhD in, that's been around for almost 60 years, has ever had an accident. At least it didn't, to my knowledge, when I was in the department. Yeah. And we've never had an accident. So I think that it's like fear mongering. Why do I feel like the gain of function research is a good thing? Because it tells you information that you would not be able to guess and know otherwise. And we've proposed certain experiments on the side as gain of function things that would be extraordinarily interesting, right?
Yep. So the term gain of function is a relatively new term. When I was training, we never used gain of function. We'd introduce mutations into genomes and study phenotypes. The term gain of function has kind of a negative connotation, right?
Well, yeah, but it was established to have a negative connotation. I mean, the person who you first used it, isn't it the guy at Rutgers who first used it, or the guy at Harvard who decided to become a virologist and a scientist, even though they're really either chemical engineers or chemical scientists or an epidemiologist, as a way to say we're sloppy and we can't be trusted. Okay. It's just like we never used the word reverse genetics before. Did you?
Never. And I was one of the first people to do it with a virus, right?
Yeah, I've never.
Yeah. Is one reason the case numbers quit dropping or the R0 appears flat due to vaccinating many who are already playing it safe with distancing, masks, telework? So, Amy, why is it plateauing? Tell us.
It's plateauing because the number of people who are protected now equals the number of people who are not protected and having the epidemics. The epidemics are now in like the 20 to 49 range. And those are the people who are not vaccinated, who decided that they no longer wanted to physical distance, mask, and blah, blah, blah. So when one population equals the other population, you get flat.
Someone asked me to turn up the volume. Let me know. I turned it up. Is this okay with you, Amy?
Sure.
Okay. Hello, it's 4 degrees in Chicago. Thanks for answering the many questions. You bet. Natalia says hello to you.
Hello.
Thank you, Abraham, for the comment on the haircut.
It's good.
You have another fan. Amy, I told you have fans. Told you.
Fans.
In a recent tweet, Rich said Sputnik was not pre-fusion stabilized. I said it was. He's right. And Amy last time said it was not. And I kind of said it was, and I'm wrong. Amy and Rich are right. I am wrong. And folks, it's okay to say you made a mistake.
It is. Unfortunately, we seem to think that that's not appropriate.
I would not get Shingrix at the same time as COVID vaccine. Shingrix is, known to have a lot of side effects, could knock you out for a day, and I would keep them separate. Amy has the ultimate background. You're right. Her background is awesome. It's a lab, and it's got colorful things. Yeah, and she works on the other side of the bay there. It's just awesome.
Well, Edmund works on this side.
That's right.
And so I work with Edmund all the time.
So Eric Topol tweeted confidently that B117 is 60% more deadly and 70% more transmissible. How can he know this with any certainty? He cannot. In fact, you know what? A manuscript just came out. I sent it to Amy today. No effect of B117 on the outbreak in England. It had no effect compared to the previous viruses. There's no difference. It was no different.
I believe that that's reproducible in New York. I believe that there's a paper that said that too. I sent you some quotes about that.
So Jeffrey, the bottom line is, it doesn't matter what initials you have after your name. It doesn't matter how experienced you are. You can make mistakes and be overconfident. And these variants are going to turn out to be, what is it that Shakespeare said? A whole lot of nothing, basically. What's the phrase? I can't remember it. Signifying nothing. Signifying nothing.
Well, I guess the guy thought when he held up the tube to his head, that's what it was saying, but it wasn't.
So my mic is too low. Some people said it's okay. I can turn it up some more, but I don't want to.
All right. So what button do I push?
No, don't touch any buttons. Don't push anything.
Fine. Don't touch any buttons. Won't touch any buttons.
But I have to fix Amy's tomorrow. I am confused. Varicella zoster. Why is it called herpes zoster when it causes shingles? Well, they're two different diseases. The first is chicken pox. The second is zoster or shingles. Two names for the same thing, and they're both different from chicken pox. It doesn't mutate. A vaccine is needed because a lot of people my age have never had chicken pox as a kid, and we're liable to get shingles. And so we get vaccinated to prevent shingles because we had the natural infection. Now, if you get the chickenpox vaccine as a kid, you're not going to get shingles. So you don't need the old person's vaccine. And Amy may even remember when I had shingles years ago.
I do remember when you had shingles. It was very painful.
Thank you, Callista. It's okay.
Hey, she's the hairdresser in California who sends us the masks.
Yeah. Yes. Amy has one of your masks. It's not bad. It's kind of like Pete Townsend haircut. That's what I always wanted to emulate, the look. Or Caesar, right? This could be Caesar, but I don't want to emulate Caesar.
Pete Townsend from The Who?
Yes, he looks like me, don't you think?
I'm not a big fan. I'm not sure.
Are there any competitive inhibitors to the binding between spike and receptors? Well, do you know of any, Amy?
So I believe that there is soluble receptor, and there have been antibodies that have been used as competitive inhibitors. But drug-wise, is he talking drug-wise?
Yeah.
I don't think that there's drugs.
It's a hard interface to block,
It's never been a successful therapy. Look at how much time and money we wasted trying to do it for HIV.
Yeah. I got the results of my T-detect test. It just says positive. Is that the T cell? Yeah, that's all they give you, unfortunately, even though it is a nucleic acid test for amplifying component of T cells. Unfortunately, that's all they give you. I'm sorry.
Well, how is a nucleic acid test going to tell you what?
I think they amplify T cell receptors.
Well, yes, but it's not going to tell you what it's directed against.
Well, it has
it's not going to tell you what the epitope is.
Well, it has to be. Otherwise, what use is it, right?
Okay, I don't understand how the test works then.
We'll have to explore that for the next episode. Does vaccine spike get expressed in the cell surface or secreted? How does this affect the immune response?
It's both. The protein gets presented on the cell surface and it also gets secreted.
And so the real effect comes when it's presented on the cell surface in an MHC molecule and it's recognized by an antigen. It's then recognized by a T cell in the lymph node, for example. An antigen-presenting cell will present it and bring it to the lymph node. A lot of people are getting their shots. That's great. That's great. George, I recently got an antibody test. I have 30 units per mil of antibodies. What does the unit mean? So it's probably an ELISA, right, Amy?
Probably.
So it's an ELISA unit, basically. And so you don't have any idea what it means unless the assay is calibrated, which they might have done, but they're not telling you. So if you can dig into the FDA documentation, you might be able to find out how it's calibrated. Like, what is 1 unit of reactivity? Which variants of concern are actually concerning, which are overhyped or not proven to cause a worse disease?
I don't think any of them are proven to cause a worse disease. I think that they're mostly overhyped, except for the ones in South Africa that seem to be immune escape to antibodies. But I'm not concerned about them because it's one epitope and you have 19 other epitopes plus all the T cell stuff.
The guy at Cornell, John Moore, I heard him quoted today, said, "None of me or my colleagues are worried about any of these, have any concern about any of these variants with respect to vaccines."
Yeah, I don't. Okay, so here's a comment. The problem with Amy's background today is that it looks like she has a convenient carrying handle on her head.
What's that?
I'm like a suitcase. I look like
monitor.
I'm a suitcase. Yeah,
no, I look like
I'm a suitcase.
All right, I'll get you some earbuds. Do you want earbuds?
Fine. No, it's fine. No, it's fine. But okay. My God, does it look like I'm a suitcase?
Thank you, Charlene, for first, what is the highest temperature any virus can survive?
Well, there are thermophilic phage that live in the sea vents.
Oh yeah, that's right. Really hot. Boiling almost, right?
Yeah. Beyond boiling, because Francis fell into them and has huge scars, like third-degree burns.
Yeah, the pressure. Yeah. Wow. But in our lab, poliovirus, you could inactivate it at what?
42.
42 Celsius, right? For a half hour?
Yeah.
Anyway, thank you, Charlene, for your super sticker. Greatly appreciated. It goes towards supporting our new studio where we will be able to make even more content.
Are we going to call it the incubator now? Like the factory?
Yes. So a listener, Anthony, said you need a name for your studio, like Andy Warhol's was the factory. And so I asked Amy, what do we do? And she said, call it the incubator. She's brilliant. Thank you, Amy.
My pleasure.
It's a great name.
Yeah, it is.
So what we're going to do, we're going to have on the door the incubator and then Microbe TV, Parasites Without Borders. What do you think of that?
Sounds good. I'm a big fan of Andy's.
My child's orthodontist treats 4 kids at the same time in one room 6 feet apart. I'm assuming the risk is pretty high of virus being passed around. Absolutely. That's not the right distancing. And I bet they're not wearing masks, right?
It is the right distancing, but because they're 6 feet apart. But I don't know what
the kids are 6 feet apart. Sorry.
Yeah. I don't know what that means. Like, is that the waiting room? Or is that the actual room where the chairs are?
Maybe the doc has, those tables that you sit on and they're 6 feet apart. I don't think so. Usually there's one table per room, right? So I don't know.
No, when I went to the orthodontist for like 50 years, there were multiple chairs. It was a large room. It was a large room. And I would say that there were 10 chairs. However, if you had one room and you had 10 chairs, you can't really be 6 feet apart, but you could put plexiglass and stuff to prevent spread like that. But I wouldn't think you should have them all at the same time. But it's hard but if it's your waiting room and all the kids are wearing masks and the parents are wearing masks, I mean, it's not the best, but I'm not that concerned about it. It's like being no different than on the subway car.
People like your name. You're the best, they say.
Oh, yeah, it's good.
Can we compare free countries to China, who has the authority to create extreme lockdowns? I think it's hard, right?
Yeah, but so for instance, Spain is a free country, but Spain and I know this because the woman who cuts my hair, her sister lives in Spain, and she said that they did not go out during the lockdowns, that their neighbors would turn them in if they went out. And all I was thinking about was it's like World War II again, when people would they would turn in their people would turn in their neighbors to the Nazis. But yeah.
West Virginia is giving out 102% of their vaccines. This is great, isn't it?
Yeah, especially for West Virginia.
What's the replication speed? Eclipse, latency, burst. Whoa. Very focused questions. Amy would know. What's the eclipse period? Do you know? Is it 8 hours?
Yeah, it's about 8 hours. Well, okay, so it depends on what cell line you're doing it in, right? Because it's going to be different in Veros compared to Calus, and then it's totally different in your body. Yeah. So it's about 8 in Veros. It's about 8 hours eclipse. Then it's pretty much done basically by 18 to 24 hours.
And and again, the burst size would depend on the cell, right?
Burst size depends on the cell, yes.
Is there cross protection from common cold coronas to SARS-CoV-2, even if slight, and if a variant can escape? It seems to me that could account for a rise in regional differences. What are your thoughts on cross reactivity with common cold coronas?
No, there is no cross reactivity, and I believe that Alexandro supported that on Sunday. He even confirmed.
Yep. Would a diet inducing ketones affect the vaccine? Ketogenic diet. I doubt it.
Not clear why.
My son tried a ketogenic diet for a while. Devin.
Yeah, I know. He was very into it. Not sure it did what he thought it was going to do, but.
Are there any great resources for people who don't understand children are not immune from COVID? I'm not aware of any. Are you, Amy?
No, I'm not really aware. I mean, maybe the CDC's website has been changed and been updated appropriately, but I'm highly doubtful since Osterholm's comments this afternoon. So no. Although they can probably talk to any parent, and any parent, like my mom, will tell them there was no way that they believed that children were not carriers or asymptomatic.
Someone needs to write a kid's book really quickly. Why is it easier to calculate R0 of outbreaks of other infections, but more difficult for SARS-CoV-2? I don't think it's difficult. It was calculated very early in the outbreak in China as 2 to 3, and that's key because that's before any interventions, before any modification of behavior. So you get the raw R0. After that, it's really hard for any virus because you don't know the contribution of human behavior. The R0 formula, and I showed it last week here, includes a variable, the duration of contact between the infected and the uninfected people. And if you don't take that into account, you don't have the right R0. And, they released some R0s in the UK. I don't believe them because they didn't include that. They only included viral factors. So it's hard to do once the interventions begin.
Well, I think the R0 was difficult to calculate for SARS-CoV-2 because we didn't take in. I think it's actually probably slightly wrong anyway because we didn't take into account any asymptomatics.
That's right. didn't.
And measles is not a good example, just like smallpox would not be a good example, because measles, everybody who gets infected develops measles rash, just like everybody who gets infected develops the pox for smallpox. So like the R naught probably is wrong for polio because so many people are asymptomatic.
Mm-hmm.
So I think it is more difficult to calculate when you totally ignore the asymptomatic contribution. And now we know it's like 60%.
Ontario is going to go into lockdown. Sorry about that. But, people probably started misbehaving. Vincent, comments on Redford's opinions on accidental leak from Wuhan lab. I think Amy was the first who alerted me to this.
I did. I was mystified by the ignorance of these comments and stuff. And he's supposed to be a well-respected individual. Someone has said he was, in a bad spot, but now he's not in that spot, and he keeps proposing it's better to be quiet and me to think you're ignorant than for you to open up your mouth and relieve all doubt.
All right, post-translation of spike mRNA. When the cell extrudes spike, why does it? Well, it's membrane bound, right? It has the transmembrane sequence, right? Right, Amy?
Yeah.
So it stays in the membrane. Like any secreted protein goes into the ER, it's transported to the plasma membrane, and then it's displayed up there. If you remove the transmembrane, then it'll be secreted, but that's not the case here. You want it to be retained in the membrane. When can we hear your interview on the street? I think he said he was putting it up Sunday. I was very impressed with him. He was really well informed, and I told him, well, good luck with your vaccine, and I hope the symptoms go away. That would be great. Thank you, Post Polio News, for your super chat. Do not some who continue to assert SARS-CoV-2 may have escaped the lab want it to be so? Maybe it absolves them of past or future responsibility. I think they're looking to blame someone. They don't have an answer because it's going to be years before we find the source. And they think this is logical. But, I work really hard to counter it, and so does Amy and many other people.
Well, it's not logical. It doesn't explain lympha's results.
Finding related viruses in Thailand, for example, right?
Right. In that 4,300 square kilometer space between, Southeast Asia and Japan doesn't explain his results. So until you can explain those results in this idea that it came from man or spilled out of a lab, you should stop. You're just showing that you didn't read. You haven't read enough, and you guys should just be quiet.
I don't understand this. And what evidence does CDC have that people don't carry? You mean after vaccination? Are they referring to the MMWR paper? Oh yeah. Here it is. Interim effectiveness
I don't know.
of BNT and mRNA in preventing infection among healthcare workers. So they have these healthcare workers who are immunized, and they let them swab themselves every week and look by PCR, RT-PCR for virus. And after 2 doses, 90% inhibition of infection. That's what it's based on, basically. But I don't think this is going to last. I think it's soon after vaccination. I think in a year you'll be infected, but you won't shed enough to transmit probably. We'll see. Hopefully we do this for next year and we talk about it. Can I do the show earlier? Amy and I will talk about it.
Earlier?
Yeah, like an hour or two earlier, because for Europe it's inconvenient, and you are almost the only source of good Q&A.
Well, 6 p.m. is not good because that's still in the middle of my work day. Sorry.
Two weeks ago, Pfizer announced that in real-time study their vaccine prevents transmission. I don't know about transmission. Do you know about transmission or just infection, Amy?
I think actually they did say transmission.
That's right.
Since the media looks at what is TRP,
I don't know.
I don't know what it is.
But this paper hasn't come out, so we like to reserve judgment until we see the data.
I don't like to make any comments. I don't like comments on press releases.
I think it was a press release.
Well, I don't comment on press releases. I don't.
Did you see the last report on Astra? 29% efficacy in preventing asymptomatic infection with B117. A bit worried for UE who relies on that. No, I didn't see the last report. I'm sorry. The last report I saw was the U.S. numbers, which were better. And I do remember in South Africa they had difficulty against their local variant. How would intact spike as opposed to spike tartar from vector RNA, mRNA be presented to T and B cells?
It's the same process.
Same process. The proteins are presented on the cell surface, the antigen presenting cell, which would then present it to T cells. If serious illness and death is only caused by our bodies overreacting, what would happen if our bodies just ignored SARS-CoV-2?
Well, lots of people don't die and they get very mild disease. The body is pretty much ignoring SARS-CoV-2.
Yeah, I don't know if it's a useful question to answer because we can't do it, right?
Can't do it, but I also don't like the phrasing of it. So our bodies just ignored SARS-CoV-2. I'm not even actually sure what ignored means in this context. So, like, ignored it, you'd be dead. You know,
yeah, I think
whatever.
They're meaning, don't make a
if you don't make a
hyper
immune response, then you just clear the virus. There's way more people who did that than got the severe disease. I'm sure Daniel has the numbers.
To see where J&J manufacturing messed up 15 million doses today. Did you hear about this, Amy?
No.
I've heard of this. And it's highly unfortunate. Please, if someone has details, let us know. Tommy Thompson's story is in my online commentary. I think it's a Virology blog. Yeah. Every country I know going into lockdown has an abysmal vaccination rate. It's very important to get this rolling out. Yes, I totally agree. I think we have to get it out more and more and more. And more states in the U.S. are lowering the age. I think New York just did, right, Amy?
They're opening it up to everyone, I think, April 15th. They lowered it.
What, 16 and up?
I think 16 and up. Although maybe I'm confusing it with California. California is definitely opening everything up 16 and above. Maybe Delaware did today. I think Arkansas. Maybe New York, he's just lowered it to 30.
Yeah, New York. My virology class said New York has lowered it too. Can leeches catch COVID? Can you use leeches as a COVID test? No, they're insects and they wouldn't be infected. So you can't use it as a test. Has no one looked at TCR sequence and correlated with all COVID antigens and natural infection? Wouldn't this address the benefits of natural vaccine immunity?
Well, I think that's part of what Alexandro and Shane do.
Yeah, you'd like to say which T cell receptor sequences correlate with the antigen, right?
Mm-hmm. It's a lot of work. You have to do tetramers, right?
Mm-hmm.
I don't think
Amy
that
has
they're
been
finished.
correct on almost everything in all these months I've followed her. There you go. There you go.
It's funny.
I'm sorry. If my volume is still low, Amy, I'll have to adjust it tomorrow. I think the big knob will lower your volume, but I don't want you to do it because then we'll have a problem. Not too much, just a tad.
Well, I played with the big knob. You tell me.
Sounds the same to me. With the
well, maybe it's not the big knob. Maybe it's the small knob.
No, the big knob is your headphone. On the mixer, there's a main knob there that can, but don't touch it. It's all right.
I don't know. There's too many knobs.
With the underreporting of COVID cases and the high rate of vaccination, aren't we near herd immunity? We did a rough back of the envelope a few weeks ago on Twitter, and you could be approaching 50%, and so we're approaching it. But it doesn't just happen overnight. It's not a switch, right? It's going to happen gradually.
Oh, it's not a switch? Oh.
It's not a switch.
I think you have to define what is herd immunity. I mean, Fauci's defining it as 80 to 90. The R0 calculation defines it as 70. I think you have to define what you want, what it is. But what do I know?
So a mix-up at a Baltimore plant manufacturing 2 COVID-19 vaccines caused 15 million doses of J&J to be ruined. How many virologists does it take to operate a mixer? Well, Amy is in New York, and I'm here, so I set up her mixer, and I can't fix it here. So, and I can do mine, but I don't want to blast mine out.
Well, the other thing is I don't know who's playing with all the knobs. I only sit here once a week. Why is everybody playing with all the knobs?
Someone may be playing with them when we're not looking. What precautions should one take when renting a small vacation home for a week?
Well, my sister just did that.
What did she do?
She and 2 friends and the dog went and rented an Airbnb for a week, and in the Catskill Mountains, and they're going with their masks and stuff, and they're having social distancing, and there's no, like, normally they would go antiquing and stuff. There's no antiquing. There's just outdoor activities. And if it rains, there's movies.
Yeah, but there was just 2 people there. Is that right? She and her friend?
3.
Were they vaccinated?
Wendy was vaccinated last Friday, and the 2 guys were vaccinated earlier. Doug is not vaccinated.
Amy, do you know why so few people are dying in Africa of COVID?
I don't think it's reported very well, personally.
I agree. Someone said here that anyone would be low volume compared to Amy. You have a very strong voice.
Okay, fine.
That's not it, though. Yeah, I think most people are low volume compared to Amy. When is Novavax submitting for EUA? No idea. But we were told a month ago, soon. Maybe any day now. Can you estimate how many more COVID-related deaths there would be if nobody was treated at the hospital? You can go onto the
that's morbid. I don't like
Hopkins
to do
site, and you could calculate. They tell you how many people have recovered. So, well, at least they used to on the Hopkins site, and so you basically can just take all of those out because all those people would have died most likely. It is morbid, but a lot of people would have died. I'm sorry I can't give you a number. What is the percentage of symptomatic infection versus total infections among 12 to 15-year-olds? Wouldn't you need to know that before claiming 100% effective against it?
No, they're just saying that they didn't see any in a certain time period compared to the placebo. That's what they're saying, and that's how they're claiming it's 100% effective. Does that
make sense to you?
It depends on how you're defining things. I mean, it's not really the right way to define it, but that's the way they're defining it. Because how are you going to find out all the people? We don't do enough testing, right? You'd have to do a test a day, and you'd have to take a chart and define very specifically what is mild disease, asymptomatic, and severe disease, right?
Yeah. Yeah.
And we don't do those things. So we do it the opposite way. Because we don't want to spend the money.
If you go to World in Data, there's a great graph, I think, looking at mortality by age. And you can just see how it just drops off in the kids, but that's not exactly your question. Okay. We are all members of Amy's Army. Amy's Army, what a great name.
I had that years ago when I had like a few people who thought that they wanted to work with me over the summer.
That's right.
I was even going to have, I was even going to have t-shirts, right?
Yes, Amy's background is real. It's not an image overlay. You're absolutely right. Neither is mine, by the way, but mine is not as cool. Amy, do you recommend any science news websites?
Science news? No, I don't read science news websites. Sorry.
Yeah, I don't either. What do you think about variants gaining interferon resistance? Do you know anything about that, Amy?
I saw the paper. I skimmed it. I'm not impressed.
And Amy is the interferon person. She knows her thing.
Yeah. I just reviewed a paper on interferon and my virus. Not so good.
Why is the WHO waffling on the lab escape hypothesis?
Actually, the guy actually now came out and said he thinks it's plausible. The head of the WHO actually now just stuck his foot in his mouth.
Yeah, I think that's wrong. It's not plausible.
Well, it's not plausible. It's just bad.
I heard a talking head on NPR called the WHO mission more of a diplomatic than a scientific mission. No, we went over. Actually, did we publish the TWIV? It's coming up.
It's coming out tonight.
Tomorrow. We discussed the report. There's a lot of data in the report. So the guy in NPR probably didn't read it. It's 100 pages. I read it, and Amy has read it, and it's good. It's not a diplomatic mission. No, that's my opinion. Do you think the same?
I don't think it's a diplomatic mission at all. I think it's just the tip of the iceberg. I think like 4 weeks and spending 2 weeks of it in quarantine is not particularly enough time to investigate all of the things. It's just like, I find it now. Oh yeah. So the WHO and, Biden's administration came out and said like, it didn't it's like a waste of space, blah, blah, blah. So the equivalent lady in China responded and she's like, oh, they should open up Fort Detrick's or whatever the one in Maryland is. And I'm like, I don't quite understand. Did not anybody take ecology and listen to like figure out where the bats are? Like the United States don't have those kind of bats here. They're not the right species. Yeah. It's just like nobody had any problem when we were talking about Zika and we said New York didn't have the right species mosquito. Like, I don't get the difference. I'm confused.
Much ado about nothing. Yeah, that's the word. That's what I was looking for.
I think that was the title of a play.
Yeah, Shakespeare. And maybe someday we'll call that a TWIV episode when we finally finish with all the variants. And yeah, it's about fitness. I taught my class that today. Fitness is what you have to.
That's why you wanted to know about G64S and stuff.
Yes. When I have questions, I go to Amy first.
Yes.
And if she doesn't know, maybe nobody does. Real world protection of mRNA vaccines. No, we don't have anything outside of the initial trial. Those people are being followed, though, and additional data are certainly being collected. That happens always for years.
No,
so
I think the health workers are supposed to be the real world.
Real world? Yeah, you're right.
Real world.
Let me show you that article again. This is the MMWR report. This is what they call real world, right? So this study, quite a few people. Yeah. But those are healthcare workers and frontline people and so forth.
And then the Israeli one that Daniel said how much he sarcastically loved the figures that I sent earlier yesterday was also theoretically real world.
I heard they were breeding bats. Were they always housed in the BSL-3? Possibly their handlers were interacting with other animals. I suppose you're talking about the Wuhan Institute for Virology. I don't
Bats
know
are
if they're
really hard to breed. You can't have
right.
bats in captivity, and you can't have these bats in captivity. There are very few. There's like 2 species of bats that are able to be in captivity. I think Tony Shouts in his TWIV discussed how rare it is, and they probably don't even breed. Go back to Tony Shouts' thing.
Tony Shouts TWIV. You have to capture them. Yeah. Where are the vaccines for the herpes pandemics? You've seen those stats lately? Yeah, there's a lot. Everybody, 90% of the population is infected. People are working on them, but they're very difficult. And, nobody has poured $24 billion into them. Maybe we would get them faster. I am such an Amy fan. I'm an Amy air conditioner. Oh, that's cool. I don't know what that means, but cool. I have to say that different type of mic. So Amy has a Heil PR40, which I used to use all the time in my office to record TWIV in the early days before the pandemic. And then when I went home, I bought this mic that I have now. And it's not as good a quality as Amy's. She's got a great mic.
It says Hilo 3. All right,
Heil
it's 8:46. And
go?
I'll do 2 more questions and then I have to add overlay.
All right. Is there any scientific basis to the popular belief that some people are genetically protected against infection?
There have been some papers that describe some SNPs in Temperus 2 and ACE, maybe.
Mm-hmm.
But you have to actually do the experiments, right? You have to build those mutations into an animal. You have to do some stuff. It's not like you just stick these mutations up to your head and say, how do you work? But I wouldn't be surprised if we find out that there are things like, what is it, the delta something of CRX-CR5 for HIV?
Mm-hmm. Yeah.
One more question.
UK variant is turning out to be something here in Ontario. In my city, hospitalizations are the highest. Our positivity has gone up. I don't think you should blame the variant. It's just the virus. It's SARS-CoV-2, and the outbreak is being driven by human behavior. It's easy to blame the variant, but that's not it. Right, Amy?
Right. All right.
Goodbye, Amy. Thank you again.
All right. See you tomorrow. Do we have anything? Oh, we have to send the MTA from the CDC to the lawyers. Please remind me. Otherwise, my viruses are not going to come. I'm not going to be so happy.
Will do. Thank you again,
right.
Amy. Good night.
Good night.
All right, everybody. It's me. I have to take out Amy here. There we go. Take out her name. Move my name down. Take this message out. Let's continue. The idea of going earlier, as you heard, Amy can't do it. I could start at, let's see, on Wednesdays, I could start at 7 and go for an hour before Amy pops on, if you want to do that, if that would help. By the way, I asked Daniel Griffin today to do a live stream with me. And I don't know when that's going to be, what time of day, but we'll publicize it. It'll be good. So we that will not take a vaccine before it is tested for many years and not for seasoned viruses, are we going to be a hate object? No, I don't think so. I think if that's what you want to do, it's fine. From my viewpoint, not everyone's going to be as sanguine about it as I am. But I don't think it's going to amount to enough of the population to make a difference. You'll be protected indirectly by everyone else. Why do antivirals that block immunity through vaccination? I'm sorry, that's the wrong one. I'll get to you. Why do antivirals that block an enzyme in the cell or substitute a nucleoside do not universally work on all viruses? Don't they all use the same enzymes? Yes. Well, they use either RNA polymerases or DNA polymerases, right? Depending on whether they're an RNA or a DNA virus. And the active sites of the enzymes are slightly different, the place where the nucleotides get polymerized. They're not all the same shape. They're very close. And so, for example, there are some nucleoside inhibitors that block the reproduction of a lot of RNA viruses, but not all of them. and they're not very potent. Same for DNA viruses. But with tweaking, I think we can get there. I think we can get a pan-RNA virus inhibitor, a pan-DNA, but there hasn't been enough money put into it so far. Why? Because for many viruses, we have really good existing antivirals, like HIV and herpes viruses and so forth. But yeah, it can be done for sure. Full immunity through vaccination looks like it blocks transmission. I presume infection when fully vaccinated is due to loss of general immunity. Now, so what I think is the following. These studies that were published in the Morbidity and Mortality Weekly Report, they show that 90% protection against infection in these individuals that were followed every week. Those people were immunized not too long ago. They have very high serum and mucosal antibody levels. Those antibody levels are going to decline in the next year. And so next year, if you did that experiment, you would find they would be infected. But the memory response would kick in 2 or 3 days. You'd have antibodies and T cells to limit the infection so you would not get disease, and you probably don't get transmission. I don't think you need to completely block infection to have herd immunity and obviously to prevent disease. That's what I think is going to happen. I think these studies are too close to the vaccination point. Because remember, you don't keep high serum levels of antibodies. You keep low levels, but you have memory cells for both antibodies and T cells. Yeah, Petite Townsend, I'd have to smash my guitar. That's a cheap guitar. I could smash it. Why don't they vaccinate adults? Because by the time you're an adult, you've been infected. The likelihood, if you're sexually active, you've been infected. I was not able to watch the whole TWIV with Alessandro. Was the question asked regarding common cold giving any relief to people? No, he said there's no cross-reactivity in the terms of protection. There are some cross-reactive epitopes, but it's not at all clear that they protect at all against disease, for example. The Pfizer, Moderna vaccine and AstraZeneca cause the same part of the spike to be made in the body. Well, the Pfizer, Moderna are both the same kind of spike. It's called prefusion. It is stabilized so it doesn't change when it's cleaved, which is part of the entry process. The AstraZeneca is not prefusion stabilized. They're 2 amino acid differences. The Moderna and Pfizer have 2 prolines inserted into the spike to lock it in the prefusion conformation, and the AstraZeneca does not. And what does that mean? You have lower levels of the spike produced, and it's less stable. It falls apart. If we're just getting to the carrying handle part, sorry, there's such a delay. Please clarify why some insist purified genome isolate does not exist. They don't, first of all, they don't know what evidence is out there. They don't read the literature. And they have an agenda. They want to say it doesn't exist so they don't have to do anything. But I can show you the paper where they pulled it out of someone's lung and they could see it infecting cells. They did an electron micrograph of it and they cloned it. Oh, that's been done many times. So I don't know why they would say that. That's a psychology question. That's not my field. Yeah, older adults, 20s I think is okay, but older adults. You know, at one point they said only women too, and then they decided to expand it to men. How do I feel about a COVID passport? I think you have your card, but that's probably, you could fake that pretty easily, right? So you'd need something more official. I don't think we're going to need it. If we get most people immunized, we're fine. So just convince everyone to be immunized so we don't have to have passports. What's my favorite immune cell type? Well, these days I'm loving T cells. I got to tell you, I really like T cells. I've learned so much from Alessandro Setes, and they're just so cool. They're the last line of defense. When antibodies fail, T cells kill infected cells. Now, they're not always pretty. You know, if you're infected with hepatitis B virus, the virus doesn't actually kill liver cells. It's the T cells that do that. So it's a bit, when it happens, when T cells work later in the infection, it's a bit troublesome. So it's not all pretty, as I said, but T cells in general are really cool. Really, really cool. Is the Bill and Melinda Gates funded study on the vaccine for use against COVID still going on, or has it been canceled? I don't know. That's a great question. We applied to the Gates. We actually sent a letter of intent. Said we want some money to make a new polio vaccine, an improved polio vaccine. They said, no. We're spending all our money on COVID right now. Come back later. Okay. They have a lot of money. I don't see why you need to do that. Could you link to the preprint that I referenced? Yeah, but I don't want to take the time to do it while I'm alone here. If Amy were talking, I could do it, but where did I see that? I'll try and do it towards the end. What are the factors that cause SARS-CoV-2 to have a lower mutation rate than influenza viruses? Okay, first of all, it's a slightly lower mutation rate because SARS-CoV-2 has a proofreading enzyme that corrects some of the mistakes that the polymerase makes. Okay, and so that makes it a bit lower than influenza virus. But they both make a lot of mistakes, and they both do variation, as we have seen in this outbreak. Did you notice Apple has added a COVID emoji? I think that's been there for a while, actually, because I've used it before. Yeah. Is it just a coincidence that the variants came from regions of the world where the testing took place? Yes, I do think. Well, I know that the UK was doing a lot of sequencing, right? And that's how they picked it up more so than anyone else. And South Africa also apparently had a great sequencing program. I'm not so sure about Brazil. I think that's a coincidence because since then, everybody else has been upping their sequencing game, and now we're finding them in other places. Yes, someone who said SARS-2 has error correction. You guys don't need me. Just come here and answer each other's questions, right? Yeah, incubator now. We're finally getting to the incubator. Okay, so that's what would be the name of the studio. It's on 8th Avenue, 2 blocks up from Penn Station, if you're familiar with the city. And you folks are welcome to come and see what I have to put together the actual recording studio, but it's my plan to make a cool thing where I could do remote podcasts or have guests come in. And every day I want to make a video of some kind to explain things. And Daniel Griffin will come and help me do things as well. Cool things are coming, I think. When I reviewed the book collection, you mentioned you found Dawkins' writing to be dated and not of interest. So it's not the writing. It's just that he was writing the book in a time before we had a lot of our current knowledge of genome evolution based on sequencing and so forth. So his idea is that it's the gene that's the object of selection, and I think that it is quite clear now. And his point is basically not an issue any longer. I would like to see it modernized, but he hasn't. Have there been other vaccines in the past to adopt the Novavax, as in recombinant nanoparticle? Not really the same, that they aggregate by the hydrophobic C-termini of the protein. The Shingrix vaccine is a glycoprotein from the shingles virus, but it doesn't aggregate like that. And then others, of course, like the HPV vaccine, the hepatitis B virus vaccine, they assemble into virus-like particles, so they're different. So no, this is the first one that I'm aware of. Beef 134. There's a podcast with Penny Moore, who's not so optimistic. Listen, the J&J vaccine prevented 100% hospitalization and death against the South African strain, B1351. And that's because T cell epitopes are not changed. So if you're not optimistic, it's because you're not thinking about the T cells, I think. Yes, I'm going to do a coronavirus Reddit AMA. That's all set. You guys want to know the date? Coronavirus, let's see. It is April 8th at 1 p.m. Eastern. Okay, that's when it's happening. Thank you for pushing that forward. And Anthony, there he is. That's the fellow who said I needed a name for the studio, just like Andy Warhol. So we went back and forth. And I told Amy, she came up with Incubator. You like Incubator, Anthony? Good. It was you who said we needed a name. I think that's a great idea. Thank you. You see, I listen. Spinning through the questions here. Osterholm says he's most worried about B117 and it says 70 to 100% more infectious. This is nonsense. He's quoting experiments that are not relevant in cell culture. And Osterholm said March would be the worst month, and March is over, and it wasn't. So take Osterholm with a grain of salt also. You could take me with a grain of salt. But you have to be skeptical. Please be skeptical. Osterholm has done many good things, and some things he's wrong about. If COVID reinfection occurs in Brazil due to P1, will the vaccine be more effective than a natural infection? I'm not sure what you mean. The vaccine be more effective than if you get vaccinated after the infection? I'm not sure. I'm sorry. Other than the rarity of accidents cited earlier, could you recap your reasons for saying the origin of SARS-CoV-2 is not from a lab leak? Okay. First of all, the closest virus to SARS-CoV-2 is from a bat. 96% genome identity. It's not the ancestor. Some other virus is the ancestor, but there's clearly something closely related. Down in Thailand, there are very closely related viruses in bats. And no one had any of these viruses growing in the lab last year. So how could they be released? And if there were an outbreak started in the lab in Wuhan, it would have been noted. And in the WHO report, they went through all the syndromic signs. They tested thousands of hospital samples, all negative. So I think we would have seen it if it had been a lab leak. But even then, I think it's, we know that viruses spill over from animals, and there's never been such a lab leak starting a pandemic. So all those reasons, very quickly, that's why I don't think it came from a lab. What are my thoughts on a single dose of Moderna? And I think you need 2 doses to get optimal immunity. In this paper, the MMWR report I just showed you, shows that you get better protection against infection with 2 doses. I'm not sure what meant fitness and variance. All right. Fitness is the ability of a virus to be maintained in a population in a host. Fitness can be affected by many things like increased reproduction, maybe transmission, antagonism of immunity, many other things. These viruses, these variants are clearly more fit because they can displace other viruses. But how they're more fit, we don't know. And people have just said they're more transmissible, but they haven't shown it because fitness can be caused by other properties, as I've said. The antagonism of immunity, the B cell epitope mutations, that could be responsible for it. So that's why I'm skeptical. And as I said, a manuscript came out showing that B1117 had no effect on the UK outbreak. No effect. It would have been the same with or without the B1117 variant. So how can it be more transmissible? In an animation, it looks like the spike contracts, drawing the virus closer. Yes. So when the spike first binds, the fusion protein inserts into the membrane. After the spike binds, it's cleaved, the fusion protein extends, it goes in the membrane, and then the spike protein hairpins, which means it folds into a hairpin and pulls the 2 membranes together. To get 2 membranes to fuse, they have to touch. It's not easy because there's water in there and you have to squeeze that out. But the hair pinning does that, and that's how they fuse. Yeah, so the hair person I go to, she has a salon with plastic sheets, but there wasn't anyone else there but her. So she keeps the density down, which is good. And I've only been, this is the second time I've been there, only in the last month or two. What's a better degree for working in virology, molecular biology or biochemistry? I think if you get a molecular biology degree, you're going to get a lot of biochemistry. So I would say that because that is modern virology, a lot of molecular biology. Thank you, Susan, for your super sticker. Very much. Every TWIV listener can indirectly influence many others. Yes, so if you learn things from TWIV, you can go and tell people and tell them what you've learned and go back to listen if they would like as well. No, you don't have to feel like an idiot. You can just keep asking and I'll keep explaining until you get it. It's fine with me. I'm a teacher at heart. Transmissibility is one part of fitness, right? It's one way that fitness can be increased. And when you look at mutations that improve a virus's spread in a host, that's an increase in fitness. So David Quammen interviewed me the other day, and we were talking about the variants. And I said, it's really about fitness. And he immediately said, oh, because fitness can be controlled by many parameters. And I said, you got it. You got it, David. David Quammen. Yeah, I mean, Dr. Griffin is going to do a live stream, but he'll probably only do it once a month at the moment. How do I keep up with the latest science? The literature, as Mark Crislip would say, right? I scan the literature. Amy sends me articles all the time, but I subscribe to RSS feeds, and I use an RSS reader to go through the articles on a daily basis, more than a daily basis, actually. I don't know what to do with Brazil. They're kind of refractory to outside help, right? They have a kind of a refractory government, I suppose. But we have a lot of people writing from Brazil and telling us about the situation. Vincent, who said they investigate frozen foods? Could have come from TV dinner. The WHO report addresses. They say there have been some outbreaks in China caused by food contamination, which I just don't. I haven't heard that. I don't see the data. I'd love to see the data. What are the most important TWIVs to listen to of 2021? Well, I would listen to the latest with Alessandro Sette. That's the first one that comes to mind. It's all about T cells and how important they are and why they're going to get us out of this pandemic. And as for the others, I'd have to look, and I want to zip through some more of these questions at the moment. So the whole AstraZeneca thing banning, so I don't know what to make of that. They've put it in the European Union, they put it in 55 million people. And, there have been some deaths, of course, and it's not clear that the clotting issues, the blood clotting issues, are a result of the vaccine or something else. Because, as you know, people do develop blood clots. In my opinion, the risk of COVID is greater than the risk of getting blood clots from the vaccine. And most of those, unless they go to your brain, can be treated, right? So I think they're being cautious, but now they don't have vaccine, right? So that's not good. Can you give blood between the first and second vaccine? I don't know. I think we've asked this of Daniel, but I don't know his answer. They will have antibodies, and it will temporarily protect the recipient, but it will wane. The antibodies go away. So if the spike is the same in all vaccines and the spike has changed in the South African Brazil variants, why would T cells still off? Because the T cell epitopes are different from the B cell epitopes, which are the ones that are changing on the spike. So the spike protein has B cell epitopes, which are recognized by antibodies, and T cell epitopes that are recognized by T cells. The T cell epitopes are not changing. That's why the T cells still protect against disease caused by the variants. Why was I frustrated when I went to a school board meeting? Because I was put in a breakout room with two other people. One of them was the school doctor who knew everything and you couldn't tell her a thing. And she knew the answers. So she was a bit, we couldn't get anything past her. I said, taking temperatures of kids is really not useful. Oh, we know that. Is really that the way to respond? But the worst part was I wanted the kids tested. And they said, no, we can't do that in New Jersey. We can't institute school testing. So I said, first of all, what the hell am I going to do here? Goodbye. I should have persisted, right? But the reality is I have to choose what I do, and I'd rather educate folks and get something done. Yeah, Rhapsody and Tea is really good. Sete was tremendous, really, really. I've heard him speak with slides, and he's not as good because, people with slides, the slides take over. But he didn't have slides, so he had to explain things, and he did really well. That's why I think TWIV is great because it makes people be clearer. By the way, if anyone is a fan of convalescent serum, Arturo Casadevall will be on TWIV Friday. And he's the guy who's been pushing that. So we'll see what he has to say. Yeah, all these countries with going into lockdowns and outbreaks, you know why that is? Because people are just going back to life as usual. Not masking, not distancing, going to restaurants. That's what's going to happen. It's human behavior. Now, it is not about the UK variant. Please do not conclude. The UK variant may be causing the cases, but if there were any other variant there, they would do the same thing. Okay, that's what happened in the UK. That's what the paper I just cited concluded. The UK variant didn't make any difference in the UK outbreak. Okay, lab leaks we got here. More lockdowns, frozen food. The WHO report suggests that they had a few outbreaks in China based on contaminated frozen food. I don't know if that's true because I've never seen the damage. Tulio de Oliveira, the head of genomic surveillance in South Africa. I'll write it down and we'll try it. Never deny accidents can happen. I don't deny that accidents happen, but there's never been an accident that has released any virus like this that's caused an outbreak. And people work with all kinds of viruses that can cause outbreak, and it doesn't happen. But the point is, Richard, every other bit of data tells us it came from nature, not from a lab. Okie dokie. What are the odds of another pandemic in the next 5 to 10 years? 100%. Probably a flu pandemic. For sure. Flu, the last pandemic was 2009. So we're getting on now to that point where we're going to have one. Thank you, Tom, for your super chat. The J&J vaccine efficacy data shows a divergence of accumulated cases starting at 2 weeks. Does this mean this is the minimum time? Yeah, I think that's minimum, 14 days, exactly. But it does improve with time after that as well, for sure. Thank you, Apollo 11 to the moon, for your super chat. Really appreciate it. How long after a vaccination should one report something as a possible side effect? Listen, I registered for that vaccine V-Safe thing after my vaccines, and I still get notifications at 6 weeks. Yesterday, I got my 6-week notification. So even up to 6 weeks, they want to know if something is going on. It's less likely it's a vaccine related, but they want to know. Yes. I was born in 1956, and my brother, who's 1 year older, was crippled by polio. What's the chance that I had the virus but asymptomatic? Very good, because only 1 in 100 infections are paralytic. So you were probably infected asymptomatically. Yep. Wisconsin opens up to the general public. Very good. That's great. Worried on the report coming from Brazil that younger people are getting very sick from P1. I don't agree that it's getting deadlier. What would make a virus get deadlier? It would get maybe more fit, right? But I don't see the data. The data I have seen is not really all that convincing. And Amy said the same thing, essentially. So I just think that the numbers are thin on the greater virulence. Is there any reason why my fully vaccinated mother is required to wear a mask when visiting her husband who is fully vaccinated in a room? No, the CDC says you can do that. Fully vaccinated people can go in rooms without masks, for sure. CDC has said that. Why is the UK variant supposedly more severe when there is pressure to be less severe? Well, that's a tough one, but as I said, I think the data are not that convincing that it is more severe. And haven't been peer reviewed yet. Thank you, Joel, for your super chat. Really appreciate it. Instead of earlier, take a few emailed questions from Europe. Yeah, you could do that. The chat only opens up 30 minutes before, but you could email. I'll set up an email for the Q&A, something like Q&A@microbe.tv, but let me make it official. Target rating point. Man, that was so long ago that I don't even remember what was going on. When can cancer patients who have recently received chemo be vaccinated? So the chemo has stopped. So if you stop it, you can resume, you become less immunosuppressed, but I don't know the time. You'd have to ask a clinician or Daniel Griffin. Bat poop is used by farmers in China as fertilizer. That's correct. Could be a source of transmission for sure. They go in the caves and harvest the guano and put it on their fields. So you can imagine, a farmer does that, gets infected, infects his family. They go into town, they infect other people. It's circulating a bit in those people, adapting better, and then they go into a bigger city and it spreads. It's a plausible scenario. So we have to find that initial set of infections. You got to do serosurveys in rural areas. I don't know offhand about vital episodes, but I'll look into some. Or if anyone else has recommendations here in the chat, that would be great. Thank you. I appreciate it. I know it's hard for people to do this at a weird hour. I am more flexible than Amy because she's doing lab work and I can come anytime. So if you want to hear me at different hours, let me know. I'll come 9 a.m. if you'd like, Eastern time. Shane Crotty was excited about Novavax. Do you know what day in April? I don't. Now here's a link to the New York Times reporting on the J&J foul-up. Good. That's a nice link for people to check out. Thank you very much. Will COVID-19 eventually mutate so the current vaccines are not effective? So it's possible that it would totally escape antibodies, in which case the T cells might have a hard time limiting it, because as Alessandro Sette said, he'd rather not have T cells. But here's the interesting thing. People without B cells, A-gamma globulinemia, they don't have more severe COVID than everyone else. So maybe antibodies are overrated. But anyway, yes, we're going to update the vaccines to match probably next year's variants, kind of like the flu vaccine on a year-to-year basis. What I would like to see is next generation vaccines with other viral proteins so we get more T cell epitopes. And that really will help a lot, I think. How many virologists does it take to operate a mixer? Well, yeah, I answered that a long time ago. What aspect of virology do you find the most fascinating? What was your most surprising revelation? I think the evolution of viruses is today we talked. I gave a lecture on evolution of viruses in my class. I think it's fascinating because we have tools now to study it, like sequencing, and questions like evolution of virulence and fitness are fascinating to me, and I really look at them in a historical way. And the current pandemic allows us to address those. I think the most surprising revelation in my career. It's got to be the first time I took a clone copy of poliovirus RNA, put it in cells, and out came virus. I was just amazed that it worked. I was amazed that it worked. I'm spinning through some of these to try and get, now I'm 3/4 of the way down. Michael, thank you for your super chat. I really appreciate it. There's a lot of chat amongst you, which is great. And a lot of repeats, of course. Again, these transmissibility and lethal numbers, they're not really based in solid data, as far as I know. Can anyone speak to the... I don't know what that means. I'm trying to get Dasak back on Twiv, but he doesn't answer. Are you concerned a bad actor will engineer a virus to do us harm? I'm not concerned, because it's really hard to get viruses to do what you want them to. And I think anyone who's not trained as a virologist can't do this. Now, maybe a disgruntled virologist, but they won't be able to do it. I think it's too hard. And that's based on 40 years of trying to do experiments. Thank you, Michael, for your super chat. Really appreciate it. Oh, what is it, the NPR? December 2019, that's my interview with him. I know, it keeps getting reach, and people say he showed that they were engineering SARS-CoV-2. People don't understand it. He said to me, I interviewed Peter Daszak in December 2019. He said, yes, we need to do these experiments where we take coronaviruses and put their spikes in viruses in the lab and see if they infect human cells. We need to know if they have pandemic potential. You do that under BSL conditions. It's a totally important experiment to do. And we did it. We knew that there were dangerous viruses out there, but nobody did anything about it. The next step would be to make antivirals, but nobody did it. Thank you very much for your thanks, especially in Italian. I really like that. My 86-year-old mother has a ITP platelet problem necessitating steroids with getting her vaccinated and then starting for antibodies to be an effective vaccine. So you're asking whether she should be vaccinated. That you need to ask your physician, because only he or she can answer. Unfortunately, Kat, I can't. All we have are informal release of data on these variants. And, it's like the viruses are in the right place at the right time. If it had been the previous isolate, you would still have an increase in cases. You're not going to convince them unless someone publishes a paper saying otherwise, and we just don't have that, unfortunately. No, the Amy's biohazard is real. All those red things in the background are real. There's no fake stuff in my lab. My daughter wanted to get vaccinated. Should she join a trial? Yeah, if you can find a trial, definitely. Definitely join a trial for sure. Why do I expect vaccine effect? I don't expect vaccine effectiveness to taper. I expect serum and mucosal antibodies and T cells to decline to low levels, steady state levels, which is what happens with every vaccine and every infection. It's just the way it is. And you have memory cells that take up the slack. You don't need to have high levels of serum antibodies. Is that clear? E protein was said to be a marker for virulence. I don't know of anyone working on it. Nope, not at all. I think the Wuhan escaped here will die when we find the progenitor in nature, like we did for SARS-CoV-1. And then it will die finally. I've heard about long-term tinnitus. We talked about that last time. It seems to be real, yeah. Here's a question we came last year. If flu cases have been so low, how will WHO decide on what will go into next year's vaccine? Don't they rely on past year's cases? They actually rely on isolation of virus, and they do have virus. So I think they'll be able to make a decision, although they'll have fewer data, yeah, but that's what they have. Could primers be designed to track variants? Sure. Yesterday we spoke with some people in New York City who are looking in the wastewater, and they use They use primers to specific parts of the genome so they can then sequence and look for variants. So you could, yeah. I mean, there's some variants that are very common in different parts of the world. You could design primers to do that. If they mess up 15 million vaccine doses, can they not mess up in a viral lab? Well, we're under a lot of regulations in the viral lab. We have and it's smaller scale. We don't have 15 million of anything. So it's harder for such accidents to occur. We have to account for everything. And there are fewer people working, which makes it easier. Why is there still a focus around herd immunity now that we have very effective vaccines? Can't we vaccinate ourselves out? Well, that's what vaccination will do. It will protect. Herd immunity is simply population level immunity. You vaccinate a fraction of the population, you decrease transmission, so the likelihood that someone else is going to acquire it is low. That's all it is. And that's how vaccines work. So why is there a focus on? Because people don't understand it, so they're talking about it all the time, and it's just something that happens. If you had Moderna, do you have an inside track when boosters come out? You mean revised vaccines? Well, they've got them already, and they can use them whenever they want. So I'm guessing that not until next year they're going to go with the current vaccines, which protect against disease. Thank you, Arthur, for your super chat. Much appreciated. Has the wingnut anti-vax movement chilled out? No, they're there. There are a bunch of people. Actually, there are just a few people who do all the noisy. But they're still there. What's the snow virus? You mean this, which arm? This one that's behind me here that I can't point to? You screen printed it? Wow. I guess I published a picture of it that you could use. Yeah. Got it. Does it matter if it escaped or happened by wild animal trade? Yeah, I think we need to know how it happened because we want to prevent it, right? I don't think it escaped the lab, but if it did, we should do something about that. And if it's animal trade, we need to know where. And stop whatever happened. Do consider searching for someone to help you. Yes, I mean, that's part of the thing that I'm going to get someone to help me. I'm looking at a PR person to help publicize. I'm going to look at a video person to help shoot and an editor. I do have a channel strip on that, Richard. I have to show her how to adjust it so she can balance with me. Is it true that people are taller and healthier? Well, the longevity has increased. The longevity in 1900 was about 50 years, and now it's over 70. And that's medicines, vaccines, public health. It's all of it together. We have 2 minutes left, roughly, but I'm going to try and slip through all these last ones here. Susan, thank you for your super sticker. I know for many of you it's a weird time of the day, and I do really appreciate that you're joining us, for sure. Crohn's disease, Pfizer, J&J, doesn't matter. Get whichever one you can get. Thank you, Access Audio, for your super chat. If we had more aggressive containment measures, would it impact the percentage of the population that needs to be vaccinated? No, because eventually you're going to release the containment, and then people will not be immune. So I think you need to get to that point where the basic in R0 R0 tells you it's 50 to 70% of the population that has to be immune, probably 70%. Virus escape maybe is it easy to hide a BSL-4 lab from public eyes? No, you go to Boston, you can see the BSL-4 lab right there, but this virus is BSL-3 anyway, which is more common. How is the adenovirus modified to produce spike? Well, some adenogenes are removed and you put the DNA in. that encodes the spike protein. B117 has ripped through every country it's landed in. Not true. Please look at the data more carefully. It's fitness, Adam. Do you understand the difference between fitness and transmissibility? Transmissibility is a subset of fitness. In California, B117 has not ripped through the population because it can't compete. It's less fit than the Cal variant. Okay? That's what I'm basing. I'm not just talking out of my head. Not my head, but, it's something else. Okay. Will your new podcast space be a replica of this homey warm one? I don't think it can be a replica, right? But I'll be here at night for these live streams, right? Because it's kind of late to be in New York City. Thank you, Martina, for your super chat. Hello, we missed you earlier on. Welcome back. And Frank has been modding for this. Thank you, Frank, for doing that. Thank you, Peter, for your super chat. All donations go to the new studio and hiring people to help me out and make better content. A lot of great questions here. Hold them for next time. Thank you, D. Wilson, for your super chat. I work in an SNF. Our long COVID seems endless and management is not adequate. Courses for clinicians are absent. Your hands are full, I know, but have you learned more from your cast? I have learned more from your cast. Thank you at TWIV, especially Dr. Griffin. Thank you so much. Greatly appreciated. And I want to thank Claudia for your super chat. Will the whole world have to be vaccinated every year? Maybe for a while, and it may be combined with a flu shot. Yeah, that's a really good idea. But I think eventually we might not have to, sort of like the common cold coronaviruses. Thank you, Couch, for your super chat. Much appreciated. I see a lot of great questions here, but it's time to go. And here we have our last super sticker from Susan. Thank you so much. So once again, folks, I am eternally grateful that you choose to come here and chat amongst yourselves and ask great questions. Yes, we're still doing Twin, for sure. And come back next week. Over 500 of you, over 200 likes. Thanks so much. See you next time. Be safe, most of all, right? Be safe.